Recall that accessory organs are organs that secrete digestive secretions to aid in the digestion of food. The first accessory organ that food encounters are the salivary glands. There are three types of salivary glands. The first salivary gland is called the parotid, followed by the submandibular and the sublingual. The salivary glands secrete a substance called saliva, and saliva is composed of serous fluid, or water, mucus, and salivary amylase. Salivary amylase digests the carbohydrate amylose. So not only is saliva important in the chemical digestion of amylose, but saliva also lubricates the food for ease of swallowing. The next accessory organ is the pancreas, which is found behind the stomach. The pancreas consists of the head, the body, and the tail. The pancreas plays two roles in two systems. It's part of the digestive system, and the pancreas is part of the endocrine system. As an endocrine gland in the endocrine system, the pancreas secretes two hormones. One is called glucagon, and the other one is called insulin. And both hormones regulate blood sugar levels, or glucose levels. In the illustration, these are the cells of the pancreas that are associated with the endocrine system. And you can see that these cells are close to or next to blood vessels. And that's because hormones are directly secreted into blood vessels. So the first type of cell within the pancreas that are part of the endocrine system are the alpha cells, which produce glucagon, and the other type of cell are the beta cells, which produce insulin. As an organ of the digestive system, the pancreas secretes a substance called pancreatic juice, which we'll talk about more in a future lecture. Pancreatic juice is produced by tiny little cells in the pancreas called Acini cells. And these Acini cells make pancreatic juice and release that pancreatic juice into passageways, which eventually lead into this large passageway here called the pancreatic duct. And from the pancreatic duct, pancreatic juice enters the small intestine. The next accessory organ is the liver, and the liver is the largest internal organ of the body. The liver is composed of lobes, and the largest lobes of the liver are the right lobe and the left lobe. And these lobes are held together by ligaments. Internal structures of the liver include the hepatic duct, which is a passageway for bile, a hepatic artery, a hepatic vein, and if you notice, there's another organ associated with the liver called the gallbladder. And the gallbladder has a duct called the cystic duct, which merges with the hepatic duct to form one duct called the common bile duct, which transports bile to the small intestine. There are 5 important functions of the liver. The first function of the liver is involved in digestion. That's because the liver secretes a substance called bile, which helps emulsify fats. And the second function is involved with excretion, eliminating waste. The third function is nutrient storage. The liver can store glucose as glycogen, can store fat, and the liver can store vitamins A, D, E, and K. The fourth function is detoxification of blood. The liver can remove harmful toxins. And the last function of the liver is the synthesis of new molecules. the liver can make new blood proteins. And the last accessory organ is the gallbladder, whose function is to store bile produced by the liver. So in the illustration, here's the gallbladder, and you can see that the gallbladder has a passageway for bile. This passageway is called the cystic duct, and you can see that the cystic duct merges with the hepatic duct of the liver to form the common bile duct, which delivers bile into the small intestine. Recall that the overall function of the digestive system is to take food and break it down into its smallest substances called macromolecules. And we want to eventually break these macromolecules into smaller units called monomers so cells can absorb them. In order to break down food, some accessory organs and some alimentary canal organs secrete digestive juices which contain enzymes that aid in chemical digestion. So again, we want to break down food into macromolecules and eventually break down the macromolecules into their smaller subunits called monomers. There are four major macromolecules that organisms need and use in a variety of ways. The first macromolecule is a protein, and its monomer is amino acid. The next type of macromolecule is a nucleic acid, and its monomer is a nucleotide. Next are the carbohydrates, and its monomer is a monosaccharide. And finally, we have the lipids, whose monomers are the fatty acids and glycerol. As mentioned to you before, enzymes are going to play a major role in the chemical digestion of food. Enzymes are biological catalysts that speed up a chemical reaction by lowering the activation energy. And activation energy is the energy needed to start a chemical reaction. In other words, enzymes will help speed up the chemical digestion of macromolecules into monomers. Some characteristics of enzymes are they're made of proteins, and enzymes are not consumed in a chemical reaction. In other words, it can be used over and over. And the last thing about enzymes is that when naming enzymes, enzymes usually end in the suffix ase, ace, such as lipase or nuclease. Looking at a graph of a chemical reaction and the amount of energy required to start that reaction, the red graph is a chemical reaction without an enzyme, and the blue graph is a chemical reaction with an enzyme. In a chemical reaction without an enzyme, you can see that the energy required to start that reaction is greater than the energy needed to start a reaction with an enzyme. As with all chemical reactions, you start off with reactants and end with products. I like to equate chemical reactions to a person rolling a ball up a hill. Which one would take longer? A person rolling a boulder up this mountain, or a person rolling a boulder up this mountain? So again, an enzyme speeds up a chemical reaction by lowering the activation energy. Organs of the alimentary canal that have digestive secretions are stomach, which produces gastric juice, and gastric juice consists of mucus, hydrochloric acid, and pepsinogen. Hydrochloric acid by itself can break the bonds of all macromolecules, but when combined with pepsinogen, hydrochloric acid and pepsinogen make a substance called pepsin, which specifically breaks down proteins. Mucus provides a protective layer so that the hydrochloric acid doesn't eat away at the stomach wall, causing an ulcer. The second alimentary canal organ that produces digestive secretions is the small intestine. And the digestive secretions that the intestine produces are peptidases, which is an enzyme that breaks down proteins, lipase, which is an enzyme that breaks down lipids, and disaccharidases, which break down carbohydrates. Examples of disaccharidases are amylase, which breaks down the sugar amylose, sucrase, which breaks down the sugar sucrose, lactase, which breaks down the sugar lactose, and maltase, which breaks down the sugar maltose. Accessory organs which produce digestive secretions are the salivary glands, which produces a substance called saliva, and saliva consists of water, or serous fluid, mucus, and salivary amylase, which again breaks down the carbohydrate amylose. Next is the liver, which produces a substance called bile, which emulsifies fat. Emulsify just means traps. So in other words, bile traps or packages fats to be digested or broken down or passed on as waste. The next accessory organ is associated with the liver, and this is called the gallbladder. And the function of the gallbladder is to store bile. And finally, the last accessory organ is the pancreas. And the pancreas produces pancreatic juice, which is a cocktail of different enzymes and substances. For example, pancreatic juice consists of bicarbonate ion, which neutralizes the acid in chyme. The pancreatic juice also contains trypsin, which aids in the digestion of proteins, chymotrypsin, which also aids in the digestion of proteins, carboxypeptidase, which aids in the digestion of, again, proteins, pancreatic amylase, which breaks down the carbohydrate amylose, pancreatic lipase, which breaks down lipids, and nucleases, which helps break down nucleic acids.